So I now switch to English, and I want to welcome Dr. Sorisik Legouillardet. She is a scientist at the National Institute for Ocean Science, IFREMER, in Nantes, in France. And she is an internationally well-known scientist, which is a specialist for viruses in food, especially for shellfish. And she has there a long-lasting experiments and focus on basic and also applied questions. She's also a specialist in new technologies for detection and for characterization of viruses. And this will be also the topic of this keynote speech. And the presentation has a title, How New Technologies Can Help to Identify Human Enteric Viruses on Coastal Environment and Shellfish. So Sorisik, the floor is yours.
Thank you very much. I appreciate it. So thank you very much, and thank you for inviting me. I'm very happy to be here, and I will try to bring you in France for a few minutes. And let's see. So you know, French people enjoy eating oyster, and of course, sometimes we are sick with oyster. So we need to know what is in our oyster. But I mean, this is not only an issue with oyster, and the introduction, I mean, really well describes the problem we have now is that the human population increase worldwide, and we need to have some clean water. I mean, the issue of the water is going to be a big issue in the next year, because we need to prepare why this is such an issue. I mean, because if you want to drink water, you can just boil it and you think you will be okay. But I mean, of course, to prepare our food, we need some clean. So we have more and more global epidemic of pathogen, and also, I mean, it's more easier to identify outbreaks now than it was before, because we have some network and we have some new possibility to identify rapidly some viruses that may be transmitted with food. So, I mean, I just download from the RAF alert report from 2021. So the notification of rapid alert for food safety. And as you can see here, I mean, the pathogenic microorganisms are still the second, in the second position. I mean, last year the pesticide residue increased a lot, but otherwise, I mean, microbes that include both bacteria and viruses are still in second and with stable numbers. So this means that, yes, we are improving. We know more what is in our food, but we still need to improve the quality of our food. So I just told you, the food can be contaminated at different steps. And of course, what's interesting us here, I mean, it's virus coming from human. And yes, they go into sewage treatment plant, but we know also that sometimes we have some failure of sewage treatment plant, and we know also that sometimes, due to heavy rain or other events, we have some overflow of the sewage treatment plant, leading to direct discharge of the virus into, for example, the coastal environment. But also we know that we can use some not correctly clean water to water the food, and so even in the field we can get some contamination. And this was demonstrated for berries or for salad or for lettuce and everything. We also know that sometimes the contamination is coming very far away, and this is really a big issue to identify. And if we want to solve the issue, for example, in coastal areas, sometimes we have to go 50, 60 kilometers away from the coastal area to find the source of the contamination. So this is really a big problem that we need to answer. So, because yes, as Raymar said, I'm working in the field since many, many years now, and I remember I just took back an old slide that we studied. It was a shellfish-borne outbreak in France following a flooding event. So we are cultivating oyster it's not a lake, but it's closed area, semi-closed area. It's like a lagoon, and we got 38 clusters. more than 200 cases identified. So that means that when you are able to identify 200 cases, it means that you have maybe 10 times more sick people. And we collected some oyster. And as you see here, some people, so here you have the stool sample results, and here the shellfish sample results. One person was infected with 7 different viruses. So he has some Aishi, astrovirus, enterovirus, rotavirus, norovirus G1, norovirus G2. So, and the point is that we almost detected all the same viruses in the oyster. So if I put this slide, it's just to show that yes, we can have a large diversity of viruses in a food, but also, I don't tell you, but we spend weeks and weeks trying to perform the PCR for all the different viruses and cloning and sequencing, so it was lots of work. So we think that, yes, it's really time to move to metagenomic for food and environmental sample, because every time we got an outbreak, so we suppose it's a norovirus or it's a astro or rotavirus, depending if there is some stools that are collected, but you don't know what is in your sample, so we need to prove and to know. So moving from molecular era to metagenomic era does not mean that we are going to completely forget the PCR targeting detection, because it allows a quantification and also it gives you some idea of really what is in your sample. But being able to analyze all the different genomes that can be detected in your food is really an important and a big issue. And so because you can get some ORs, you can investigate all the genomic content, you get both a culturable and non-culturable microorganisms. So of course this is really important also for bacteria. And you know, but that's a big issue also. Sometimes, and I will show you, we got so many sequences we cannot identify, so the interpretation still needs a lot of work. So, but anyway, the metagenomic has the potential to advance our knowledge in a wide variety of fields. So I just summarize the 2 parts of my topics here by presenting the 2 methods we are working on now. So what we can call the meta-barcoding approach, so it's like a targeting metagenomic. So it's useful to detect one type of virus, so you will amplify because you still begin to do a PCR. So you begin to do a PCR, you amplify the region of interest, then you prepare the library, then you map what you got, and you are able to identify and to genotype the diversity of the virus, of this type of virus you have in your sample, and you can say, okay, I have genotype A or B. But what could be more interesting, of course, is the metagenomics approach. Either direct or for virus, what we find important is to insert a step of selection or enrichment of the virus sequence. Otherwise, you just cut the DNA or RNA, you prepare the library, and then after the sequencing, you perform a de novo or reference-based assembly, and you get some partial or full genome if your sample is really contaminated, and then you are able to identify the different strains, to describe the diversity, and even to discover some new virus or at least new sequence within the genogroups or the virus type you are looking for. And also it's very interesting to compare with the clinical sample, and this will help also for food outbreak analysis. So just first few words on the amplicon-based metagenomics. And so this is really very useful for norovirus. So most of you know what's a norovirus. It's a major public health issue. It's the main agent of acute gastroenteritis in humans, and I think we begin to see an outbreak in France, not yet really, but it's beginning to appear. So it's really, for now, the pathogen more frequently implicated in foodborne outbreaks. It's really a large number of foodborne outbreaks linked to norovirus. There is a genetic sensitivity to this virus, so there is several strains circulating in the population at the same time. And it's a very stable virus, so we can find them in sewage and then in water and then possibly in our food. And because it's a very short segment, RNA, 7.5 kb, high genetic diversity, there is a high number of recombinants. So if you want to type your norovirus, you need to identify one portion in the capsid region, here the VP1 typing region, and in the polymerase typing region. So this has been admitted since many years, and it was just the classification had just been clarified 4 years ago now. So the good point with this approach, targeting amplicon-based metagenomic, is that you can use the ISO method. You have your extract, so you perform the PCR, you know it's positive for norovirus, then you say, okay, it could be interesting to describe all the diversity of the virus. You just keep your nucleic acid. You perform the reverse transcription, and on your cDNA, you perform the PCR, either for the capsid or the polymerase. If your sample is really presented a very low contamination, you can even perform a nested PCR, but just after you need to consider this step. You get your amplicon, you tag them, you perform the mice. For example, this is what we are doing usually in my lab, the mice sex sequencing, and then we can use some frog pipeline bioinformatic analysis, because that's a big issue also for people who were used to perform PCR. We are not used to bioinformatics. So we can use a frog suite, tool suite that is really, was developed for bacteria on 16S or 18S, and there is different clustering, swarm, remove chimera, and filters, and try to clean the data. Then you get your sequence, and then after you can just type with database. So first, just to be sure that this method could be useful, we compare on oysters that were implicated in outbreak. We compare, I will say, the old method by doing a PCR on the capsid, cloning with and Sanger sequencing, and we sequence 8 clones per sample. This is 5 samples. One sample was contaminated with 2 strains. This sample get many strains. I think it was 4 different strains with a different number of clones that were positive for this strain. And we, exactly on the same extract, we performed the amplicon-based metagenomic approach. And as you can see here, I mean, from 4 different sequences here detected by cloning, we were able to identify 9 different sequences in this sample. So yes, this approach allows, and it's more, I would say, easy when you begin to perform that, to perform that, the cloning and everything. It's less task. So then we just apply also this approach to oysters that were sampled during a big study around Europe. So we selected 200 samples of this study, and we look for the norovirus, and we tried to we applied this amplicon-based metagenomic. So we identified quite a large number of G1 polymerase and VP1 region, and for G2 and polymerase and VP1 region 2. What can be useful by using and applying this tool to such big, large number of samples is to study the evolution and the presence of different norovirus strains. So, for example, here at the beginning, so this is G1, and I just put here the VP1 typing because it was too. So, at the beginning, so for the first, it was a 2-year study. We cut it in 4 blocks. So November, the first winter, the G1.2 was majority detected in the sample, but disappeared around the study, while the G1.1, that was not really frequently detected at the beginning, was really increasing at the end of the study. So we can see some evolution. The same for G2 also, the G22 that was frequently detected in the sample at the beginning of the study was still present, but was less frequently detected, while the G24 here that was not so frequently detected at the beginning of the study was really more frequently detected at the end of the study. And maybe also it can help us to better understand the behavior of the norovirus during summer, because it looks like we have less diversity during summer. So this is type of studies that can be useful to perform with this type of approach. But of course, the goal is to develop and really succeed to use metagenomics. So again, I mean, we try to be simple. We use the ISO method, but then after, so you can do your PCR, it's positive, you say, okay, this sample seems to be interesting. Yes, you recover the leftover, the proteinase is supernatant, and we found that it was interested to sonicate, just to elute more virus, PEG concentration. We tried to eliminate as much as possible the free nucleic acid, RNA or DNA, and then after we extract the viral RNA. Then we prepare the library, and what was important to detect human enteric viruses is to insert enrichment step, and we use a kit that was available on the market, but unfortunately, they stopped to produce it. It was some probes that targeted vertebrate viruses. So it selects all the different vertebrate viruses. So then after you just wash, so it allowed to eliminate some interested sequence. Then after, again, the big work begins. I mean, it's a pipeline bioinformatics. So you get your raw FASTQ files, then there is a step of quality filtering sequence just to deduplicate some sequences are really amplified a lot, and you may also eliminate some bacteria, or for example, for shellfish, we eliminate also all the sequences that belong to oysters just to try to be clean. So you got your clean FASTQ file, then you begin the assembly step. So there is some you can use some computer script. Then you get your context step. Then you begin the mapping, the taxonomic assignation, and then after you really try to compare the different sequence you obtain. And I'm sorry, it's written in French. And for each tax ID that was selected, you can either choose to a FASTA or BLAST file. So you really are able to identify your sequence. And then after, we also set up, like we begin to insert some quality control or criteria in our laboratory, because we say, okay, if it's shorter than 500 bases, it's too complicated and it's too, it's difficult to be confident in the results. And also, we also decided to set up a criteria of 100 reads, just to say, yes, there is no reason. I mean, if there is really low number of reads, maybe it's something that is it's not real sequence. Then, of course, hopefully for norovirus and different type of virus, we have some database. But so for norovirus, hepatitis A and E, and enterovirus, for rotavirus also. And there is a tool also that can be easily used. Is a genome detective. You just enter your sequence, and it gives you the name of the virus. So of course, after we always check that it's really that is really a good identification. So by doing that, and this is some example we obtained with 3 different oyster samples, so 2 Ostrea gigas and 1 Ostrea edulis, we were able to detect some Reoviridae, Picorna viridae sequence, Picobirna viridae. We always find a lot of the sequence in environmental sample, Calici viridae, and Astroviridae, that is also very frequently detected in environmental sample. So this is for norovirus detection here. So our 3 samples, and we were able to identify long contigs here, G2P7, G26, some G1 also, and here we were able to obtain 2 complete genomes, G2.6P7 and G1.2P2. So, I mean, yes, really, I mean, by doing different steps, I mean, we can identify with good confidence some norovirus sequence. But then after, we just want also try to increase our accuracy and say, okay, what we are looking for. And so we begin to compare different approaches, like the technical replicate. So at the step of the performing the the library, we repeat it 3 times. So this means that for each sample, we prepare 3 different libraries. And yeah, we can get some different results depending on the library. First, sometimes there is some library did not provide any sequence of interest. This is for norovirus polymerase at the top, VP1 at the bottom. So yes, sometimes library, we don't know why. Either it's failed, but we got other sequence, virus sequence, but not the norovirus sequence. And then after, each replicate represents a variety. But globally, more or less, we always find the same sequence. So this is interesting also to see. And maybe we can merge all the sequence we obtain so we can increase our confidence in the results. Another approach that we are trying, and we are finishing our paper, is to do biological replicates. So instead of performing the extraction only one on a replicate, you perform 4 replicates. And then all the 4 replicates follow exactly the same extraction. We perform 4 library, 4 sequencing. And for sample 1 here, again, this is for norovirus. It's the number of contigs we obtained for norovirus. 1 replicate did not give any contig for norovirus, while the second library provides more than 30 contig. But if we added all the different reads and so analyze again the different reads all together, we really increase the number of contigs for norovirus because, of course, we increase the number of data related to this sample. For example, but at the difference, I mean, for this sample, all different libraries give quite the same number of contigs, and when we merge all the different data from these 4 libraries, we did not increase so much the number of contigs, because this means that more or less all those contigs were more or less the same, they were identical, so you just got more confidence, but not so much diversity. And so finally, and this will be the last part of my talk, I mean, because we know, and so shellfish can be, of course, be a food, but we think that they can be also a sentinel because they are filtering everything that is in the water. So, of course, when you grow shellfish as a food, there is some area of production, but we know that in all the different countries there is some wild shellfish that can filter some human pathogen, but also some virus that can circulate in the different animals. And so we, with a team at the University of Yaoundé, we performed a study, and a student, a PhD, Patrice Bonny, I mean, he collected some clams, and he analyzed them, and we selected 22 samples, and we performed some metagenomic approach using the triplicates of library for each sample. And then we performed Illumina NovaSeq sequencing. And this is what we obtained for this virus. So even if we try to eliminate as much as possible the DNA, we still detect some DNA virus, especially the Circoviridae. We got plenty of unclassified Riboviridae, maybe probably lots of failed sequences that have not been really identified. But we were happy to find some Picornaviridae here in all samples, Picobirnaviridae, not so much here, Caliciviridae also, and Astroviridae. And we detect also plenty of other sequences. So if we look to more precisely to 4 families that may infect human, Astroviridae, Caliciviridae, Picobirnaviridae, Picornaviridae, you see that we find, especially for Astroviridae, plenty of unspecified sequence. I mean, it looks like mainly mammastrovirus, but we cannot identify them precisely. Other mammals or bats. This also was really for Caliciviridae. We find plenty of sequences that have been previously detected as virus-infected bats. I mean, I have no proof that they really are pathogen for bats. and Picobirna viridae also, and Picorna viridae we find from sequence from reptilia, from bird, from other mammals and bat. And if we look more precisely for norovirus, this is a sequence we find for the human norovirus, G26P6 and G2.2BE. And we also detect lots of sequence similar to bat calicivirus A10. So this is really something that is interesting to follow up and to see, because when you eat a sample, so for now, there is no risk because in the population locally consumes those clams, but boil and smoke and everything, so probably the virus are destroyed, but you never know what can happen. So just some points of discussion, and for sure it's not a conclusion, because I hope you understand that this is not done yet. We are still working on it, but we are improving. The targeted metagenomic, it's sensitive and easy to apply method to describe diversity of one type of virus genus. If you want to look for norovirus, it's really a good tool. Metagenomic, it's a promising approach to describe the viral diversity. But environmental and food sample are very complex to analyze, considering the low concentration and microorganism diversity. So we still need to improve the method, but we are keep looking a way to enrich the sequence, because otherwise it's very difficult to get long contig. We are still working on trying to improve the approach of technical or biological replicates that increase the sensitivity, allow longer contigs, and get more confidence in the results. And of course, we also, as for many other approaches, using molecular biology, quality criteria are very important. And applied to shellfish, whether considered as a food or environmental sentinel, this method will help to a better understanding of some zoonotic transmission events. And with that, I will thank you very much, and thanks all the people who contribute to this work. Thank you very much. Happy to take questions.